The overall goal of this procedure is to extract all the surfactants from environmental samples, such as atmospheric aerosols, and determine their concentrations and surface tension curves.
This method can answer key questions about surfactant concentration and surface tension behavior in all the relevant fields. The advantage of this technique, in particular the extraction step, is that it isolates surfactant from complex matrices for characterization.
The implications of this technique extend toward the understanding of cloud formation in the atmosphere because surfactants are expected to favor the transformation of atmospheric aerosols into cloud droplets.
While this method can provide insight into surfactant in atmospheric aerosol, it can also be applied to other systems such as soil samples or microbial cultures.
So the idea to develop this method came from the lack of a selective extraction for surfactants in atmospheric samples. Until now, only water extraction has been used.
First, immerse a previously prepared filter sample in ultra-pure water for 2 hours at 6 degrees Celsius in a closed glass petri dish. Every 30 minutes, shake the petri dish while holding it flat. After removing the sample from the refrigerator, filter it with a clean syringe filter and add the filtered solution to a pre-weighed 60 milliliter glass bottle. Following this, rinse the petri dish with 5 milliliters of ultra-pure water. Then, filter the water with the syringe filter and add it to the solution in the 60 milliliter glass bottle.